Hi, a warm welcome to you all to this free public EACR webinar. My name's Andrew Binns. I'm the membership manager at EACR. I'm just going to briefly introduce the session and then I'll manage things behind the scenes and during the Q&A. The EACR is proud to be one of the partners of the EUCanImage project. This 5-year project closes in the coming weeks, so this is a great time to find out more about its aims, achievements and impacts. We've got 4 presentations from key contributors with an interactive Q&A to finish. You can ask questions at any time through the Q&A box, but we won't be answering these until the end of all the talks. The Q&A box is usually on your control panel next to your video and mic, and depending on your device, it could be at the top or the bottom of the screen, and it could also be hiding in the More section where you've got the 3 dots. We'd like this to be an interactive session, so there will be an opportunity to ask your questions verbally. And when we do the Q&A, I'll prompt you to open your mic and you can do that. You can also upvote other people's questions that you'd like to be answered. And we won't be using the raise hand function in this webinar, so questions through the Q&A, please. The webinar is going to be recorded. We'll add it to the members' resources for the EACR members to access. And we'll also provide a link to them on members, and we aim to have that in place next week. So it's my pleasure to introduce our first speaker, Maciej Bobowicz, who will start with an overview of the project. Maciej graduated from the Medical University of Gdańsk in Poland in 2006, and after a 2-year foundation programme for junior doctors in the UK, he returned to Poland to complete extensive general surgery and surgical oncology training. And that was back at Gdańsk again. He focused on breast cancer diagnostics and treatment, and in 2020 was appointed assistant professor at the Second Department of Radiology to lead his first work package in the EU, this project, the EUCanImage project. He's the enthusiast of the responsible use of AI in medicine, particularly cancer imaging, with interest in artificial intelligence in oncology, radiomics, bioinformatics, process automation, precise medicine and oncology, and new cancer diagnostics and treatment technologies. Maciej is a board member at the clinical working group of the AI for Health Imaging Network, a leader of the work project in RadioVal project, and a task leader in the European Federation for Cancer Images, the EUCAIM project. So Maciej, please do share your presentation, and the floor is yours.
Fantastic. Thank you very much, Andrew, for this kind presentation and introduction. I just have too many presentations on my desktop, as I can see. Just one second. Okay, can you see my slides? Okay, I take it for yes.
Perfect.
Fantastic, thank you. So we are here today to talk about EUCanImage project. EUCanImage project stands for European Cancer Imaging Platform linked to biological and health data for next generation artificial intelligence and precision medicine in oncology. I'm talking here on behalf of the clinical working group and our project coordinator, Professor Karim Lekadir, and I will briefly tell you some details why we started this project, because I think that's where we should begin this workshop really. So we started to write the proposal in 2019. So ages ago, almost 6 years, exactly 6 years ago, we applied for funding for this project. At that time, as you can imagine, AI world was not that advanced. We didn't have ChatGPT at the moment. We didn't have multiple multimodal AI agents. At the time, we had only a handful of AI algorithms that were FDA cleared, less than 50 of them. Today, we have more than 1,000. So it's a completely new world and completely different world after 6 years of EUCanImage project. What was important at that time, we had a huge promise from AI tools that could help us in daily practice for radiology, oncology, and surgical oncology. So when we thought about this project, at that time, there was just one huge repository for imaging data coming from states, so the Cancer Imaging Archive, with partners in our project as well, but there was no such a repository in Europe on large scale with multiple data sets, well curated with images and enriched with clinical information. And that was our idea, to build a trustworthy AI-driven European cancer imaging platform. And that's what we did. So let's start the actual presentation. You know, when we think about repository with multiple subjects, you have to start slightly smaller. So we needed to concentrate on some cancers, probably one of the more prevalent ones. We knew that there are already some repositories working on lung cancer, which is the most common cancer in human, but there were very few of them working on breast cancer, the most prevalent in females, colorectal cancer, and liver cancer. And that's really what we wanted to explore at that moment. So we decided to build data platforms and capabilities that will address real-world unmet clinical needs for cancer cases in 7 different use cases, which I will talk about slightly later today. To achieve such a very ambitious goal, we needed a proper consortium. On the right-hand side of your screens, you can see Professor Karim Lekadir from Department of Mathematics of Barcelona University, who is coordinating this grant. To build the consortium, we needed really several pieces of this puzzle. So we needed some research infrastructures, we needed some academia for AI and infrastructure as well. We needed some industry partners to help us out with the tools and platforms that were needed as building blocks. We needed some data, so we needed also some knowledge and experience in oncology and radiology. So we involved 5 clinical sites. Later on, we built it up. We added 2 more clinical sites. We needed organizations such as European Association for Cancer Research, who is hosting us today. We also involved European Society of Oncological Imaging and European Institute for Biomedical Imaging Research, who are helping us in here today as well. So as you can see, it's pretty large consortium of 20 partners coming from 11 European countries and the United States, where we included the Cancer Imaging Archive to help us shape this very important European initiative. When you think about the platform for imaging data and for cancer, you certainly need something that will cater different needs. So we needed to serve clinical sites to provide the data. We needed some central infrastructures to host the data, and we needed some solutions for AI developers. It looks so simple, 3 different building blocks, but when you think about it in more detail, you need slightly more complex design, and that's what you can see in here. When we move forward, all those partners need some contractual framework, because in modern day with GDPR compliance or HIPAA compliance in the United States, you can't really shift the data from the data origin, so patients or hospitals who are hosting their data, to central repositories or AI developers. So you really need a proper contractual framework. So we need, again, to serve data curation from clinical sites. Then we need to look after data transfer to central repositories and have some legal agreements in there. And we also need some legal agreements for data usage by technology partners and external partners who will have access to our data from this year on. And again, it looks simple, but when you think about the details, there are multiple different agreements in between single partners participating in this proposal. And at the moment, we are still closing up the data sharing agreement, so we can share patients' data with final data users from outside of the consortium through developed EUCanImage platform. It's all based on our EUCanImage portal that was developed during the last 5 years, where you can explore the data, you can access AI tools, you can submit the data to our portal, you can get some support, and you can, of course, get some information about the consortium and about the whole project. It connects all the clinical centers, AI-enriched research, and it has tools for data discovery. So what are the building blocks in here again? So certainly when you have some data in a platform, and today we have a lot of data in our platform, you certainly need a catalog. So to explore EUCanImage datasets, you need the data access committee portal to be able to apply for the data and to receive agreements and data sharing agreements, just to mention one. To provide imaging data, you probably as a data user would like to have this data not in only raw format. You probably would love to have some annotations, segmentations, some radiology reports, and that is possible to one of our partners, Collective Minds, who provided annotation suite and developed it further to cater the needs of EUCanImage project. We also have AI VRE, so virtual research environment, as a development platform. We have a partner who developed in silico trials platform, also available in here for in silico validation of AI algorithms. And we have OpenEbench, which is part of Elixir benchmarking platform. In here, you have just a small snapshot of our metadata catalog. It's based on use case number 7, which I will talk about slightly later. Again, data from my center, Medical University in Gdańsk. You can see my name as the head PI and contact. And as you can see, it has a very rich number of different metadata tags, frequently not available from other data sets. In here, you can access it. You can look through multiple different metadata parts. I told you already about the Collective Minds radiology annotation platform. So as you can see in here, it's annotation suite available to all researchers worldwide. You can see the dashboard that was developed specifically for full field digital mammography, 4 views of the breast, right and left, CC and MLO images. It allows us semi-automatic annotation of all the lesions in the breast, and the classification, it can contain specific reports like BIRATS classification or ACR density scoring. It can contain also specific fields for research which we used and are available also in the data platform. When you think about images and non-imaging data that enriches the data set, you certainly need quality controls. And that's what we did. We implemented 2 different quality control systems. One of them is more human-based. It's visual assessment based on image quality score like Likert. It's 5 points score, and it was judged by the clinicians on the clinical utility of images. On the other side, we have automatic quantitative quality score based on the PIC score ranges, and it's also attached to every patient and available for all the data sets. In here, you can see the flow for liver cancer use case, which is our first use case. In here, we talk about the primary hepatocellular carcinoma and early detection of those cancers. So you can see the outline how the pipelines can work and how we approach this topic. When we move further, I told you already that we have virtual research environment. In here, you can see tools available for breast. So when you log in into the virtual research environment, what it allows you is to use the data that is already available through our platform. But not only, you can upload your own data. You can upload the data sets that you have rights to. And then you can use our ML and the deep learning tools for mass detection, mass segmentation, prediction of malignancy, analysis of radiomics, and estimation of treatment response. We've got also ML toolbox in there. So it's very useful for designing AI tools as well as testing them. At the end, all of us, when we develop AI tools, want to assess them just to see if they are fit to practice, if we can get the good benchmark with those tools. And that's available in the OpenEbench. And that was my first presentation. Now I will move to a second presentation. So just let me stop sharing. If you have any questions, please put them in Q&A and we will answer them at the end. Okay, so let's share a second one. So now I will tell you slightly more about the use cases that we used to develop this platform and to test it, because the use cases were, on one hand side, a reason for the whole research. On the other side, we use them to validate the whole platform to see if it works, if the data that we collected and our ideas can work in more explorative way. So when we think about the use cases, altogether, we worked on 7 use cases. You can see they are numbered from 1 to 8. That's a bit strange, and you can't see really use case 2. Use case 2 was also in the proposal, but it occurred undoable based on legal agreements between different parties. So we couldn't really reach the needed numbers for any AI development. And we managed to convince European Commission and our project officer that it's basically not possible. We had to move some data to other use cases to provide exactly the same numbers of patients, but still manage to deal with 7 different use cases. The first use case is on liver primary cancers. Second, the third, fourth, and fifth use case concentrate on colorectal cancer and metastasis, and cases from 6 to 8 were used to develop tools for breast cancer based on full-field digital mammography and magnetic resonance. So let's go slightly into more detail. We defined use cases as really clinical questions. So the first use case on liver primary cancer, the definition was if AI could increase the diagnostic sensitivity of liver MRI for detecting small hepatocellular carcinoma lesions smaller than 20 millimeters in cirrhotic livers while keeping the specificity high. So if you're working with HCC, you're pretty well aware that detection rates for lesions smaller than 20 millimeters are slightly smaller than for the bigger lesions. And literature gives you the number of 60%. So detection rates of around 60% for nodules smaller than 20 millimeters, and then you have to classify them properly. So you have to know if it is HCC or something different in the liver of the patient. In here, we decided to go for 3 different tasks to achieve this goal. So we needed automatic detection of liver lesions, we needed automatic segmentation of liver lesions, and then we wanted the patient classified as having hepatocellular carcinoma lesion in their liver or other lesions, so non-HCC lesions. To do it, we used a liver MRI scans with contrast enhanced at the time of the used at the time of the diagnosis to make the diagnosis. The whole dataset was enriched with multiple clinical parameters as well as the ground truths. Ground truths in here are twofold. The first of them is histopathological verification of the lesions done during the biopsy or excision if it was performed. The other way of verification was long-term observation of those patients and clinical information that those lesions didn't develop any further. So we knew that it is most probably not HCC because the patient didn't develop the disease any further. So clinical diagnosis, currently also often used in many countries for differentiation. Now we move to use case number 3, which is colorectal cancer use case. In here, we decided to answer the question if AI could help us identify liver metastases in colorectal cancer from the preoperative CTs. The rationale in here is that the occurrence of colorectal liver metastasis is a negative predictive factor for overall survival. And we all know that the overall CT sensitivity for colorectal metastasis is around 80-82%, with pretty low specificity of 72%. And here again, we wanted an algorithm that will help us automate the detection, allow segmentation, so it will show doctors, radiologists where the lesions are, and it will help you to classify automatically into colorectal liver metastases versus other lesions. Sorry, it's a mistake in here in the slide. To do it, we certainly needed preoperative contrast enhanced CTs, loads of them, and I will show you the numbers at the end. Also in here, you can see on the right-hand side some images. One is the pre-treatment liver CT with no lesions marked. The other one is with the automated outline of the lesion, and it's kind of like orangey yellowish, right? So you can see it in the right lobe of the liver that there is a lesion of around 2 centimeters picked up by the AI system. We move to the next colorectal use case. In here, we decided to go for a pretty tough task for radiologists, but also for AI. Why is it tough? You will hear later on, but lymph nodes in pelvis are kind of difficult to spot if they are small, and they're pretty difficult to differentiate between metastases and reactive lymph nodes. So the ones that tell you about the infection but contain no cancer cells inside. Again, rationale in here was to improve accuracy for lymph node detection and improve sensitivity. As you can see in here, positive predictive value for radiologists is pretty low. It's just around 50%. So it's like tossing a coin and guessing what you have in the image. So again, in here, 3 tasks: automatic detection, automatic segmentation of those lymph nodes, and classification, metastatic lymph nodes versus non-metastatic. And as you can see in the images below, you can see ADC map with ground truths marked by the radiologists, and you can see a use case, the same patient done by AI algorithms. So you can see the segmentations done by AI system. Use case number 5 is even more difficult than this one, because in here we are trying to achieve something not possible for the radiologists so far. So we asked the question if AI could help us predict level of response to neoadjuvant radiochemotherapy based on pre-treatment MRI in rectal cancer used for local staging and restaging. Rationale in here again was that at that time, 2019, and really today, we don't have really good tools to predict the response level after neoadjuvant treatment in rectal cancer. And we would like to predict it even before we started this treatment, which makes it even more difficult because it will be based just on a single study from before the treatment starts, and it is MRI that was used for staging. The idea behind it was that we would love to improve treatment allocation in those patients. So if we knew that the patient will respond to neoadjuvant treatment, we would still offer it. If we had patients that we will know that they will not have a complete pathological response, then we will need to adequately allocate another treatment not to lose time in those patients. The task in here is pretty obvious. We need an automatic classification. It's twofold. Or you have pathological complete response or you don't. So pretty straightforward. And then we used pre-treatment pelvic diffused weighted contrast enhanced MRIs. Use case number 7, slightly not in order, sorry for that, but it is very similar. It just refers to breast cancer. So in here we are again exploring possibility of using AI algorithms to enable the escalation of neoadjuvant systemic therapy in patients highly likely to achieve pathological complete response. Why this? Well, I told you already, it's the most frequent cancer in ladies, and we really have a lot of these patients, and a lot of them are treated with neoadjuvant treatment a priori. So just to remind you, use case number 7, optimizing treatment planning in triple-negative breast cancer and HER2-positive breast cancers. These patients, a lot of them require neoadjuvant treatment. If they are locally advanced or if they have triple-negative breast cancer, even if the tumor is so small as 1 centimeter, we know that they will have some benefit from neoadjuvant treatment. The difficulty in here that we discussed with Lidia, who will be the next speaker, is that in these patients, we never know who will answer and if the response will be complete or not. And it changes everything, really. So if I have a patient as a clinician who comes to me and I know it's triple-negative breast cancer, and I know it's bigger than 1 centimeter or I have the lymph nodes, I certainly would offer them treatment nowadays. But I would really love to know which of them is in this around 47% of patients who will respond, or is in the other group who doesn't respond to this treatment. And as I told you, we don't know at the moment who will respond for certain. So if AI tools could help us with this, that would be exactly perfect. For this, we again used pre-treatment breast diffusion contrast enhanced MRIs images enriched with multiple data, multiple clinical variables. What happened here? Okay. Use case number 8. Again, breast cancer, but in here, very simple task. But this task equates to millions of studies every year. In here, we concentrated on screening, mammography screening. As you know, millions of ladies every year get screening done in the screening centers, and then somebody has to assess these images. And it depends where you live. If you live in Europe, we usually have double reading. It means that two radiologists independently review every single mammography from each patient, and they have to agree. If they agree, that's perfect. If not, then there is or supervisor or they discuss and agree. So it's a time-consuming process. And when you think about millions of studies, two readers, that's a lot of radiologists having to work on those images, a lot of hours spent reviewing mammographies. And there's even something more interesting in it. When you think about the occurrence and prevalence of cancer in those images, it's striking because less than 1.5 to 7%, depending on the publications, of screening images contain some lesions, and not all of them are cancers. So a lot of radiologists will be spending time reviewing completely normal mammography images. And that's good for the patients, of course, but bad for the health systems, because it means that there's a lot of radiologists reviewing normal studies and having limited time for those ones that contain some lesions. So we tried to develop a system. In 2019, there were quite few available tools for that task. Today, we have quite a few more available commercially. You can buy pretty good tools, but at that time, there weren't many. There were just 3 companies that had it FDA cleared and they could sell it, but there was not a lot of evidence it worked in clinical practice. So we concentrated on automatic detection, automatic segmentation, and classification. In here, we have 3 different classes. So the studies could be normal, benign, or malignant. Pretty straightforward. And at this stage, we just needed to sort out normal studies from other studies and let concentrate radiologists on benign and malignant, differentiate them. On the right-hand side, you can see the small snapshot from breast imaging reporting and database system scoring. So BIGR scoring developed by the American Society of Radiology. As you can see, there are multiple classes, right? And class number 4 is even further subdivided to A, B, and C. And all of those lesions for A, B, C, and 5 are suspicious. And how do you filter them out as a radiologist? Multiple factors that you need to take into consideration. Here, you use AI tool, screen out, and just mark all normal studies to be excluded from the review. That was the idea. Subcohort, like half of the patients in use case number 8 are cancer patients that we provided. And in here we again went very, very ambitious because we thought that if nowadays you need to do the breast biopsy in all those recalled patients that have some lesion in the breast, then you have to do a very extensive pathology workup just to receive simple information, what subtype of cancer it is. Because nowadays we use molecular subtypes. There are 5 distinct molecular subtypes of breast cancer. As you can see on the right-hand side, luminal A, be like HER2 negative, HER2 positive, HER2 enriched, and triple negative breast cancer. So this information allows you proper allocation of patients to treatment. So if we were able to use AI to develop such a system that will be very automatic, we could omit breast biopsies because we not only know that the patient has breast cancer, but we also know which subtype, and we will be able to allocate them to correct treatment. So very ambitious task and a very difficult task. But that's something that is still to be developed. We are still working on this last task, and I think, Lidia, you have something in your slides to talk about it. But to do it, we had to do a lot of work. I told you we have this annotation platform from Collective Minds, used for data annotation. It's very good because you can do pixel-wise segmentations for index lesion. Index lesion in our vocabulary in the project means the major lesion that was biopsied. For other lesions, we used bounding boxes just to show them to the system so it wouldn't get too many incorrect scores when looking for lesions. We segmented and annotated multiple pathologies. We have cancer, we have benign lesions, we have microcalcifications. We also scored BIRATs, ACR density. We have information on molecular subtypes from pathology reports. We have all the information and annotations for foreign bodies. So a lot of information that can be used in future research. And as I mentioned, we supplement it with a lot of clinical variables. In here, we call it clinical requirements. We defined clinical variables needed for different use cases. You can see that for use case on treatment response prediction, we had like 56 different variables, limited at the end to 39. So there's at least 39 parameters for every single patient, containing things like response level, but also treatment they received, because that's often lacking in such database, because you can't really predict treatment response if you don't know what your patient received. And in here, we provided exact dosing, treatment schemes, and medication, oncotoxic drugs which patient received. On left-hand side, you can see a table, and in here you've got use case number 1, so hepatocellular carcinoma, and multiple information points that we provided for these patients. As you can see, it was the simplest one. Still, it's a lot of information to receive for AI development. So what we ended up with is over 20,000 patients, 30,576. Just checked it last week, especially for you. You can see that the data is coming from 5 different countries. We have GUMET, which is Medical University of Gdańsk, Poland, FCRB, which is representing Spain. We have Kaunas Clinics from Lithuania, UNIPI from Italy, so University Pisa, UMIA University from Lund, and we have UB, which is providing also data from Barcelona, from Barcelona Clinic. So you can see the spread of the numbers based per clinical site, and you can also see how many patients you have available in different cohorts for different use cases. And as I mentioned, screening cohorts are usually the biggest, the easiest to reach. That's why we have there over 12,000 patients in this screening cohort. I think I have to end here because I'm already over the time. I'm sorry for that. And to get to another point, you can see in here very lovely picture of very small portion of our consortium working on this use cases and working on EUCanImage project. I would like to thank everybody who was involved, and I would like to introduce Lidia, who is next speaker. Lidia is a 5th-year PhD candidate and senior AI researcher at the University of Barcelona with extensive experience in computer vision, deep learning applied to medical imaging. She co-leads the AI working group in our EU-funded EUCanImage project. She organized the International Mamma Mia Challenge on breast cancer AI. Very interesting challenge. You can look it up on the internet. Really cool. It's all about the treatment response to treatment, so use case number 7. She is also serving as the organizing committee member for MICAI 2025 Open Data Initiative. Within the EUCanImage, her research focuses on developing AI models for cancer diagnosis and treatment response prediction. Her expertise includes domain generalization, multi-center data harmonization, and ethical, trustworthy AI in healthcare. Lidia, the floor is yours.
Thank you very much, Maciej. Everything is correct?
It is. Thank you.
Fantastic. So I will be explaining the general approach we follow in EUCanImage for the AI development of all the use cases presented by Maciej. So now I will start with the simple question, or not so simple, that what it takes to develop AI in a healthcare scenario. So we will need to define the tasks first, which are linked to the use cases and the clinical questions. We will need to prepare the data. We need to design the models that can be AI or machine learning models, train them and validate them, add some kind of explainability, so these models are easy to understand for clinicians, experts, and also patients. and run an in silico validation of this model. So we try to achieve this during the 5 years of EUCanImage. And of course, the data was being added in the repository at the same time. So we had to start with public repositories. Yeah, sorry, I just saw the message
So
to
we can
share my
see you now.
video. Yeah, thank you. Well, thank you. And yes, so I will continue. I will go step by step in more detail. So the task definition. I'm not going to repeat this, but we have 3 different use cases, one for liver, one for colorectal, and one for breast. To solve each one of these clinical questions, we need to develop some AI models. So for use case 1, first we need to segment the lesions, the HCC lesions in liver MRI. We need to classify them, running a binary classification, HCC and non-HCC. And maybe we need to run a radiomics analysis to add some explainability, have more details, explanations of what is going on in the lesion. The same for colorectal use cases. So we have CT, we have MRI, and we need to also detect the lesions and classify them. In this case, metastasis versus non-metastatic lesions. And for the breast cancer use cases, as Maciej explained in detail, we have screening, which is sorting the mammograms, benign, normal, malignant. But then we have more in-depth analysis for treatment response in MRI and also in mammography to try to differentiate between the tumor subtypes. Okay, so in this case, the breast cancer use cases also is the same. We need to detect or segment the lesions automatically. We need to, in this case, run multi-label or multi-class classification models. It can be to detect the 5 tumor subtypes, or if we want to detect just triple negative, we can run a binary classification too. And then a 3-class classification for normal, benign, or malignant lesions in mammography. And on top, in each one of these cases, we can run the radiomics analysis. So as you can see, this ends up in being a lot of different AI models, and we need experts in each one of these fields. So for that, we had radiomics working in liver use cases. We had Siemens focusing on colorectal, and then UV that we focused in breast cancer. And altogether, Maastricht was working in radiomics and explainability of these models. So then I'm going to go to the data preparation. For data preparation, first we need to download the imaging data from XNAT repository, right? Where the data is hosted, and where now in the next session we will see more in depth how it works. So we download the data that it's in DICOM and the images and extract the annotations, for example. Then we will transform this data for a more user-friendly, to a more user-friendly format, which can be nifty, PNG, or with the image format that the model needs. We need to download the clinical data that in EUCanImage, part of it is in XAMPP, but we also have an independent repository for clinical data. So we download it and we adapt it to the AI model, and then we filter depending on the task. Not all the patients can be used. Unfortunately, for each one of the use cases, we need to do a bit of data cleaning. So this ends up in having an AI-ready data set. So this is an example, but an AI model needs a really well-structured imaging and annotation data. So all the folders should follow the same structure, same number or similar number of images with naming, standardizing the naming. And then we need a CSV with inputs and outputs that the model expects. Okay, then the model design and validation. Model design and validation, I will go through the model that briefly introduce Maciej, because we don't have time to go through each one of the use cases, but we are happy to solve your questions later on. So for lesion segmentation, normally, well, it's widely known the NN unit. We use a 3D automatic segmentation network based on unit that it's able to segment HCC lesions. So we will use these automatically segmented HCC lesions to later on, like next in a cascade, train a classification. In this case, can be a ResNet as an example, that will give us a probability of this lesion being HCC or non-HCC. So the basic idea could be if this probability is low, and with enough confidence, we will discard this lesion. If it's high, we will require an extended radiological evaluation. But then we go to performance, and we need to measure the metrics on an external validation set. So our clinical question was if we can keep sensitivity and if we can keep specificity while having a good sensitivity or recall. What we can see here is that we have a really nice sensitivity, but the specificity is still low. So we still need the radiological input in this case. But the potential application of this model, for example, of this model that it's been trained, it's not the final model, but the current model, is that it can help to triage patients because it really does well the HCC lesions. Then adding explainability. So I'm going to explain why explainability is so important. So these AI models, of course, are black boxes, and they need to somehow, we need to transform these probabilities or these attention maps, etc., to something that is understandable or that is comprehensive for a human, or that it's not expert in AI. So it's also really important because there are ethical considerations. It helps to identify errors and biases. It improves the decision making in the clinical environment, and also because of regulatory compliance. So when we started the project, we did a review, gathering all the methods that were developed at that time. And you can see it was 2022. But this review was done before, so a lot of things have changed. Now we have foundational models, etc. But by that time, we gathered all the methods that related to explainability. And then we built a toolbox. So this toolbox, as Maciej showed, it can be found in our virtual environment, accessible through EUCanImage platform, and it englobes many different explainability approaches, visual, statistical, etc. And then last but not least, the in silico validation. So once we have this model, we added some explainability, we would like to get some feedback from clinicians, right? So the same Maastricht has this live platform that you can register and that helps to visualize the cases, add some explainability graphs on top, and gathers the feedback from clinicians to validate these tools. And then I would like to announce, I would like to give the information of all the publications in the consortium. They are live on the website, and the research results. There are many to list, but I will focus on two. One that is the radiomics quality scores version 2. So the first radiomics quality score paper has already over 5,000 citations. So it's been really impactful. Many people use radiomics now. And then Philippe Lab made another iteration on top, and then published this radiomics quality score this year. If you want to have more information, they have this really nice website that guides you through the radiomics readiness levels and provides many different information. Okay, and last, the future guidelines. So all this experience, all this consortium, all the members, the inputs from all the consortium members were built these future AI guidelines. That means fair, universal, traceable, usable, robust, and explainable. That made the word future, and it's a really nice paper that with guidelines to follow in AI. This is the QR code. I really think it's a must-read paper, and that's all for the AI part. I'm going to stop sharing my screen, if that's okay. I hope the audio was nice. So now it's my time to introduce Stefan Klein, which I'm really sorry because I haven't prepared your introduction. So I will let you introduce myself because I completely forgot. I'm really sorry, Stefan.
No problem. Thank you very much. Your words. And also, I think we win a bit of time by being quick on that. So thank you, Lidia, for a very nice presentation. I'm going to share my presentation. All right. So yes, my name is Stefan Klein. I'm a professor in applied medical image analysis at the Erasmus Medical Center in Rotterdam, the Netherlands. And I'm the chair of the Biomedical Imaging Group Rotterdam. We are a research group doing research on medical image analysis using AI. But in this project, we were responsible for the data platform, so the technical infrastructure that enables the research. So I would like to take this opportunity to give you a bit more details on the platform and the metadata catalog. So I'm going to start with this slide that Maciej showed in the beginning with his presentation, showing with the overall project where clinical sites put data in a central infrastructure, and then AI developers, as was just shown by Lidia, get the data from the central infrastructure and develop the AI models. And then there's this very detailed underlying architecture. So actually, I would like to zoom in a bit in detail on some components of this architecture. I'm going to start with the central part, the central infrastructure where we store the data. So the main question is actually, so how do we store this imaging data of more than 25,000 patients in a fair and standardized manner? So fair means findable, accessible, interoperable, and reusable. So our strategy was to not invent the wheel ourselves, but make use of existing research infrastructures that already have proven solutions for each type of data that we are dealing with. And by making use of these existing research infrastructures, we also get a kind of sustainable hosting. So because EUCanImage is a project, so there's a beginning and an end, but these research infrastructures, they typically have a more long-term horizon. So to give an example, to explain it in more detail, let's start with the imaging data, which is the most important part of this project. So for the imaging data, we are using the Bioimaging XNAT. That's hosted actually by Erasmus MC, so our group, but I will explain you that bit more detail. So what is Bioimaging? So Bioimaging is the European landmark research infrastructure for biological and biomedical imaging. It's part of this European Strategy Forum on Research Infrastructures called ESFRI. So it's a really large consortium with many countries involved. But in practice, what it is it's actually a collection of nodes who offer specific research infrastructure services. One of these nodes is the population imaging flagship node Rotterdam that we are hosting. And that developed because we were working a lot on large population studies, and for that we needed infrastructure. And now we have developed that, we want to offer that to others. So one of the core pieces of infrastructure that we are offering is the imaging data repository. So we are hosting an XNAT platform for storage, sharing, and management of the medical imaging data, mainly focused on DICOM data. And this has been running live since 2014 already. It contains data from more than 250 projects, often multi-center studies like EUCanImage. And it contains data of around 60,000 subjects. And we have been using this successfully in projects like EUCanImage, also Dutch national projects, other European projects you can share, also the currently running EUCaImage, that's also at the backbone. It's hosted by a professional company, so since it's sensitive data, there's a help desk for first-line support, and we have some system for the contractual agreements. So what is IXNOT? So IXNOT, it's a software that provides functionality for data visualization, uploading, download data, so organizing it and sharing it with others. So it has some mechanisms for access control. There's some search and exploration functionality, and you can link to it to do processing or even do build in some processing pipelines. So it's an open source software. It's not developed by us. It's actually developed in the US by a group, but there's an active community around it, and it's already sustained also for quite some years. And many groups around the world are using it. A key feature is that it imposes a structure to your data. So it's a very straightforward structure. It organizes your data into projects. Each project has multiple subjects. Each subject contains different experiments or scan imaging sessions, basically. And each experiment contains all the scans that are acquired then. And there's a fine-grained access control, so you can, on the project level and user level, say, okay, you have read-only, write, or you can read and write rights. It's mainly targeting on DICOM data, so imaging in DICOM format, but also it's flexible. You can also store non-DICOM data in it, so it's quite flexible for research in that way. And lastly, there's a graphical user interface, so it's a website, but there's also an API, so you can script against it. And we have developed a Python library that you can easily interface with and automate tasks. How does it look? Here's an example for one of the use cases. So this is the screenshot. So from the main project page with a short description. And then there's a list of all the subjects that you're seeing with some limited clinical variables that are stored here, but not the full list. I will come to that later. And then within each subject, you can see, or within each imaging session, you see here a list of the scans. At the bottom, you see maybe some familiar MRI sequences that you can see here. Then you can click Manage Files, and there you can really see the files that are stored, and you can also, so the different DICOM files or some annotations that are stored here in just a common folder structure. You can also download the images via the website if you have the access rights, of course. And also, as I said, there's an API, so you can automatically write a script to download them using the IXNOT toolbox, and it's an open source tool to interface. Right, so that's about imaging data. So now for the non-imaging data, and so the clinical data, we have another solution. IXNOT does provide some basic functionality for that, but actually there are solutions that are more powerful for that. So we opted for the European Genome-Phenome Archive as a basis for storing the non-imaging data. This is a bit of an overview. It contains a core. At the core is actually the choice to how do you standardize your clinical data. So and we have chosen to adopt the FHIR model, so FHIR. That's that you see at the right. So that's the way we want to represent the data in the end. So the platform provides basically a kind of transformation pipeline to get from the input data to this desired format in which we store the data. And it includes some conversion tools and also a data entry tool. That's a RedCap at the bottom. So this FHIR schema, so that's the data model. Let's not go into detail it, but it's actually just a very formal description. Okay, for each variable, what are the possible values, using which vocabulary are they described? So that's established for all the use cases. And then for the data entry in this REDCap system, we have designed this eCRF forms where the data can be entered. Right, summary of this part. So we have, we now include more than 25,000 patients in this platform. The data stored following this FAIR principles. So it's accessible via different ways and stored according to, in a standard organized way. We use the standards like DICOM, FHIR, and also SNOMED and LOINC vocabularies. It's based on this existing research infrastructure. So for the non-imaging data, the EGA, and for the imaging data, the Eurobioimaging IXNAT. So it uses, the main tools are IXNAT, REDCap, and EGA. And I didn't talk about this due to the time, but it supports centralized but also federated analysis. So next question now that I want to spend a few minutes on is how to find the data for people outside this consortium and see if it's suitable for your study, because you don't have access to this data already. So what we needed is a kind of a catalog. So I'm going to talk about one part in this, which is in the top of this infrastructure flowchart, which is the Imaging Catalog. This catalog is also hosted by EIB, which is also an institute for biomedical imaging research that exists after the project. And in this catalog, that stores only metadata of each data set. So not the actual data, but only descriptions of, of what kind of data can you expect to find there. So it's actually a bridge between the data owners and the data users. Within this descriptions also we include links to the actual storage platforms, so IXNAT for the imaging data and EGA for the clinical data. And we have developed a metadata model again to describe what kind of metadata we want then to expose in this catalog. So the components, it's pretty straightforward. So there's a metadata model at the core. Then there's like an interface to fill in this description of your data set. And there's an interface for the data user where they can search among all the data sets in this catalog. There's in each description, there's what I said, links to the imaging and the non-imaging data storage, so that once you identify the data set, that you can see where it is stored. A bit more detail on the metadata model, although let's not discuss it in very much detail. So there's actually a structure. It's a metadata model based on an existing standard for this kind of catalog models. So there's different levels. There's an imaging biobank, and then that contains multiple image collections, and then there's raw data sets, but also processed data sets and annotated data sets. And then in each category, there's a number of variables that you can enter. Just one concrete example for a data set that we released also as part of EUCanImage is the Erasmus glioma database. So it's not part of use cases, but also kind of a bonus data set. So here you show a description, and then you can see that this data set contains a number of like original data sets. So T1 weighted images, T2 weighted flare images, T2 weighted spin echo, and T1 weighted contrast enhanced images. And then on the right bottom, you see that it also contains some segmentation. So and on this level, it is described, and there's a link to the publication here. And also there's a link to where the data is stored. And here you see an example that Maciej already showed for the use cases, where we actually did more effort also in describing in full detail the whole data set, and describing what data is available in each data set. Just as a note, so what I said, there's an interface here to provide the metadata information based on what the data owners basically know, but that's a bit of handwork. So they have to type it in, and that's not a very sustainable strategy, you could say, because when we change the data input, we always have to call again the data providers, please update this description. So what we're now working on in a follow-up project in EUCaImage is to automate this whole process, so to extract this metadata actually from the original data sources. But that's just a side note for future work. So as a summary, for the metadata catalog is the main tool to make data findable. So this is the F from FAIR. The first requirement for FAIR. At the basis is a metadata model that describes what kind of metadata we describe for each imaging collection. It's based on, again, open source software. It's a package called Malgenus, developed in Groningen in the north of Netherlands. And there's in the catalog we have links to the data storage platforms where the actual data is stored. Currently, it relies on manual input, but we're working on automated sync via this FAIR data points. Let me see. Yes, this was my talk. Thank you for your attention, and I'm giving the word back to Andrew to moderate the Q&A session. So I'm looking forward to your questions.
Thank you, Stefan, and thank you to all our speakers for taking us through this really, hugely impressive and complex project. I can see that it's generated some thoughts and some questions for the discussion. I'm aware of the time and everybody's heard quite a lot of information. So if there's anything that they want to ask, I guess please do that now. Click on that Q&A box. And if you've left your name, we can get in touch and let you talk to the speakers and perhaps have a follow-up. But what I can do is start with the questions that we have and that we've kind of a little bit answered in writing. So I think the first question that we came through was have you achieved all the objectives for all the use cases? I think, Maciej, that was directed towards you.
Yeah. So yes and no. Well, we managed to achieve all the objectives for 7 use cases out of 8. And as I mentioned in the beginning, we had to abandon use case number 2, which was concentrating on refining LIRADS. I'm not sure if you are familiar with this scoring. It's similar to BIRADS. It only refers to liver lesions. And we decided when we wrote the proposal to refine LIRADS classes like 2 to 4 to make it more specific, like binary system. Apparently, to do it, you really need a lot of imaging. And our all clinical sites were unable to provide as much data as we would need to answer this question. So we decided to basically cross it off because it wasn't possible in 5 years with limited resources inside the project. I hope it answers the question.
I'm just going to head back to the open questions. There is one from Anisha, and I'll see if Anisha can ask her question. Their question, sorry. They're able to.
Okay, I think
open the mic.
Yeah, I think I can try to answer it because I'm the only clinician on the panel. So I understand it refers to staging of the cancer, right? If you could do it with AI. I think you could do it, probably not today. You can do it in the future. We were trying to work on such tools, but it's like multi-agentic systems. You can't really do it with a single AI agent to make staging. Staging requires a lot of imaging combined together. So you need multimodal data to provide staging for cancer. When we talk about the ctDNA, that's slightly different. And I've seen some studies that were trying to achieve it. So probably nowadays you could do it. It's just outside of the scope of our project.
I hope that answers that question. I think it probably does. The question at the top of the list is an anonymous one again. It's how did you manage the anonymization of imaging data and transfer to the platform?
Stefan, can you handle it?
I could answer to that. Yeah, that's a good question. I didn't cover that in the presentation. I had some backup slides that I could show, but I think we're a bit short on time. But we have actually a quite a elaborate ingestion and annotation pipeline. Let me share it anyway. Then I can use my backup slides. So actually, that's one of the bottom components that I didn't talk about. That's hosted by a company, Collective Minds Radiology. So we are not reliant on this company. So from Imaging, we also provide an anonymization pipeline based on an open source software, clinical trial processor. But in this project, it was convenient to use the service of this company. And they were a partner in this project. And basically we defined the steps. So where partners, they upload the data to this well, let's show this. I think this slide is better. So basically from the local clinical archive, they did a pseudonymization using the tools. And and that's all happening within the clinical private network. Then the patient ID is replaced by a pseudonymization ID, and then the data is stored to is transferred to the Collective Minds platform. And then actually we had a script running, a connection between the two platforms. So actually within the Collective Minds platform, we also use the annotation capabilities for doing segmentations. And then once that was done, we moved the data from Collective Minds to Ixnat using a script at the backend, basically, that was developed in collaboration with them.
I think there is an additional very important point, Stefan, that during this upload, we are doing also auditing of the anonymization, which was very, very important for all the clinical sites.
Yeah, good addition. Yeah. And actually we did another re-anonymization also, like from the platform to our platform, just to make sure no mistakes are made there. Yeah.
Excellent. The last one that I have that's open is, how do you see the relation between the EUCanImage and the other cancer initiatives like the EUCaIME?
Yeah, I could take that as well. So in EUCaIME actually is, so we are also a partner in EUCaIME, I think many of us actually. And the EUCanImage project has in a way become, I would say, adopted in the EUCaIME project. So, and this is a great opportunity because it provides us also with a kind of sustainability strategy for the future, that the work we have done is not lost. I already mentioned one example where we are gonna also work on automating this synchronization of the catalog. So which is also a goal in EUCaIME. So also EUCaIME has this catalog, so we're also now aligning these catalogs. We have aligned the metadata models. So there's some ongoing work in EUCaIME that will actually provide more functionality for also analysis. So I think there's a good alignment. That's and so I'm really happy with the EUCaIME project now, which is actually continuing on an even larger scale, combining the efforts of other projects as well.
Well, thank you for those questions. I can't see another question open, and we've answered a few more in the detail that people will be able to see those answers as well. We are pretty much bang on time for the session, which is very good. So I guess it's time to thank everybody for those participants and for you guys for your time and for these questions at the end. I think I'd like to thank Maciej, Lidia, and Stefan for taking the time, and also Ignacio and Jamilia, who are supporting in the background that no one's seen, but they're working hard. And obviously this is the final few weeks of the project, so I'm sure that there's still many ends to tie up still. So a good time to have this, but also not completely finished, I'm sure. A little bit of housekeeping before we finish, and we'll send around a short survey to all our participants in the next couple of days. Which would just like their feedback, which helps us to improve sort of our events. And also we might get some ideas about future topics and themes for some of these sessions too. As I said before, we'll make a recording of the session available, and members and non-members will have links and ways to access that. From an EACR point of view, we do run monthly webinars and regular networking sessions. So a reminder to people to read their bulletins and have a look at the website to see upcoming opportunities and things that they can attend. So yeah, that's all from me. Thank you again to everybody. If there's anything anybody would like to add, please do that, and then we can finish the session.
Thank you very much.
Thanks a lot, everyone.
Thank you, everyone.
No problem. Okay, thank you all, and enjoy the rest of your days. Thank you.
Thank you. Bye-bye.